Spermatogenesis is one of the most difficult topic of embryology. Today I will try to make it very simple for you. Hello, I am Dr. Aizaz from medicovisual.com. Welcome to this visual lecture. So as we have discussed previously that sperm and ovum, they combine through fertilization to form zygote. And you know sperm has how many chromosomes? 23, and ovum has how many chromosomes?
23.
23. So 23 plus 23, they will combine to form a 46 chromosome zygote that is having 46 chromosomes, right? And through subsequent division from zygote, a complete organism will be formed, and that complete organism, that complete human, each of his or her cells will have 46 chromosomes, right? So here you can see the number of chromosomes remains same. The reason is that the cell division that is occurring here from zygote onwards, that is what? That is mitosis, right? And then when this human will mature and he or she will form the sperm or ovum from the germ cells, then the number of chromosomes will be half, that is it will become 23. So what we are discussing today is gametogenesis. What are gametes? Sperm is a gamete as well as ovum is the gamete. Sperm is male gamete, ovum is female gamete. So the process of production of male or female gametes, or you can simply say the process of production of gametes, is called gametogenesis, right? And gametogenesis of, naturally it is of two types because gametes are of two types. So it is spermatogenesis, when male gametes or sperms are formed, and it is called oogenesis, when ovum, or I mean female gametes, are formed. So first we will discuss today spermatogenesis, and in subsequent lecture we will discuss oogenesis. And remember, both of these are types of gametogenesis, right? Clear up till now? So let's move forward. So where does the sperms are formed? You know, they are formed in testes, and in testes, before the baby is born, before the baby boy is born, of course sperms are formed in boys, not in girls, you know this. So before a boy is born, in his testes, primordial germ cells will migrate. They will come from primitive streak all the way through the yolk sac into the testes. I know these terms are quite ambiguous for you. Don't worry about that. We will discuss that in detail in when we will talk about sexual development. When we will talk about development of male reproductive system. So primordial germ cells that have migrated from primitive streak into the developing testes of the male fetus, what it will do, it will ultimately form mature sperm. So from primordial germ cells to a multi-step pathway in adult male, or in the male that is that has just gone into puberty, it will form mature sperms. So how it will happen, we will discuss that in detail. But remember, the primordial germ cell cannot directly transform into mature ovum. Why they cannot directly transform into mature ovum? The reason is that they are not well equipped, they are not well prepared to undergo meiosis. Of course, you know this process requires meiosis because it is a reduction division. From primordial germ cells has 46 chromosomes. From 46 chromosomes we have to form sperm that have 23 chromosomes. So of course process of meiosis will occur here, right? So primordial germ cell cannot directly go into meiosis. They will first form a cell that is well prepared, that is well equipped, that is ready to undergo meiosis, and that cell that is ready to undergo meiosis, we call it primary spermatocyte. So first primordial germ cell, through a multi-step pathway, it will form primary spermatocyte, and then primary spermatocyte through multi-step pathway will ultimately form mature sperm. Remember, mature sperm is also called spermatozoa. This term zoa mean animal. So why they call it sperm animal? Is it an animal? No, it is not an animal. Actually, the sperm, it has a tail. It can move on its own. It has its own energy synthesizing machinery. So probably scientists initially thought that it is just like an independent animal. So they named it spermatozoa, right? So here again, the primordial germ cell, after it will form primary spermatocyte, this primary spermatocyte will first undergo meiosis 1. It will first undergo meiosis 1 and it will form secondary spermatocyte, and then secondary spermatocyte will undergo meiosis 2 to form spermatid. And now you can see this spermatid does not look like sperm. Its nucleus is same as that of spermatozoa, that is 23 chromosome haploid, right? But it does not look like spermatozoa. So finally it will undergo cytodifferentiation. It will undergo cytodifferentiation to form mature sperm, right? Now let's look at this process in a bit more detail. So this is the cross section of testes, right? It is the cross section of a mature testes of a mature human, an adult male. And testes have this convoluted tubular structure. What is this? This is called seminiferous tubules. So before a male, after a male is born, a male baby is born, up till puberty, up till before puberty, just before puberty, seminiferous tubules is not a tubular structure. What it is a solid, you can say it is a solid tube. It is not a hollow structure. It is a solid structure, and in that solid tube there are Sertoli cells, right, along with the primordial germ cells that I have discussed previously. But before puberty starts, just before puberty, the seminiferous tubules, they mature, right? Overall the testes mature, they increase in size. We will not discuss in detail, but what happens to seminiferous tubules is that a lumen appears inside that, right, inside the seminiferous tubules, and Sertoli cells that were first randomly arranged into the solid structure, they are arranged into periphery, right, and there are special pits that are formed, special grooves, special recesses that are formed into these Sertoli cells, and these primordial germ cells are fitted into those recesses. Let's look this in a 3D software in Blender, so we will learn it in a bit more detail. So here you can see we have, let's remove this. So you can see these grooves are there. Can you see these grooves, these recesses, these pits inside the Sertoli cells? And these Sertoli cells, they are arranged in the periphery, on the periphery of Sertoli cells, right? And then in these grooves, these primordial germ cells are arranged. Can you see these, right? Primordial germ cells, they are present in these grooves, in these recesses. And what is this greenish structure? Actually what happens that these Sertoli cells, they are tightly packed with each other, tightly, closely packed with each other, and you can see there are tight junctions in between the neighboring cells. So this is one Sertoli cell and this is the other Sertoli cells. In between them there are tight junctions. This is called blood testis barrier. Why there is this blood testis barrier, we will discuss that later, right? So let's move forward. So what happens? Let's look at in a simple 2D diagram. So what is this? This is the this is special recesses or pits or grooves of Sertoli cells, and in that grooves there is primordial germ cell that is lying, right? Before puberty start, this primordial germ cells, some books even mention that before the baby is born, these primordial germ cells, all of them, they are transformed into two types of cell. Two type of cells, we call them spermatogonia. Actually, they are transformed into two types of spermatogonia, right? What are these spermatogonia? You can see one spermatogonia is dark and the other spermatogonia is pale. So pale spermatogonia and dark spermatogonia. I know this is confusing for you. Let's make it very simple, that what these pale and dark spermatogonia means. Or they are also called spermatogonial stem cells. So these pale spermatogonia, they undergo mitotic division, simple cell division, and they will form two types of cell. One cell is the same spermatogonia, right? One cell is this same very same spermatogonia, and the other cell that it forms, it is destined to form mature sperm. It will ultimately form mature sperm. So what is so unique about this cell division? This is asymmetric cell division. Normally what happens, that let's suppose this is a cell and it will divide into two cells, right? And these two cells in which it will divide, they are very different from this parent cell, right? These two cells, they are different from this parent cell. But here something unique is going on. Here it will divide into one cell that is different from parent cell, while the other cell that is very similar or same as that of parent cell. So what is the use of this cell that is same as parent cell? Basically the reason is that as the sperm, billions of sperms are released in each ejaculate, right? So what will happen, that so we need millions of sperms, rather billions of sperms, to be formed all the time. Throughout the life, maybe zillions of sperms are formed. So to form that much sperms, if this thing was not there, if this ability was not there, this self-replicating ability, self-renewal ability rather I should say, if this self-renewal ability was not there, very soon all of these primordial germ cell would have used up, right? In an adult male, all of these primordial germ cell would have used up to form mature sperms, and then in subsequent ejaculate there will be no sperm and person may have become infertile. So that should not happen. That is why these nature has provided these cells with the ability to undergo self-renewal, that they will form one cell that is destined to form mature sperm, and the other cell that is formed that will maintain the population of these parent cells. This type of cell division, it looks like a stem of a plant, a stem of a tree. So we call them stem cells. These are stem cells, right? These are undifferentiated cells and these are stem cells. We also call them spermatogonial stem cells, and both of these are Spermatogonia A, and this cell which I am talking about right now is Spermatogonia A Pale or Dark? Pale. Pale. Spermatogonia A Pale. Now the question arises that what these Spermatogonia A Dark is doing. They are capable of cell division, but they do not usually divide. They rarely undergo cell division. Now why I am saying they rarely undergo cell division, although they can? Well, the basic aim of a species, a basic aim of nature is that the species should survive, and to make the species survive, it should have a remarkable ability to reproduce itself, right? So nature has provided these reserve cells. What are these reserve cells? For example, let's suppose if due to some cytotoxic reagent or radiation, these Spermatogonia A Pale, these are destroyed. Most of their population, let's say if most of their population is lost, what will happen that these dark Spermatogonia A Dark cell, then they will come into action and they will divide into one. They will also go asymmetric cell division, undergo asymmetric cell division. They will form one Spermatogonia A Pale and one Spermatogonia A Dark to maintain its own population as well, right? So look at the beauty of nature, that it has provided a reserve mechanism as well, that let's suppose if these cells are lost, their population is lost, these dark ones, they can divide to resume the population, to restore the population, right? So this is the difference between Spermatogonia A Pale and Dark. Now the question arises that why these are pale and these are dark? Well, these most likely the reason is that these Spermatogonia A Dark, their nucleus is more tightly packed, right? Their nucleus is more, their DNA is more condensed, because they do not frequently undergo mitosis or cell division. Their nucleus is more packed, while these that routinely undergo mitosis, their nucleus is less tightly packed, it is more loosely arranged. That is why it appears pale on microscope, and this appears dark on microscope, right? Now how this Spermatogonia A will start to transform into, what is this, mature sperm. We will discuss just now that what happens, that there are certain growth factors that are released from Sertoli cells when puberty starts, and with the start of puberty, when these growth factors that are released from Sertoli cells, they will cause this maturation of spermatogonia into mature sperm. Spermatogenesis start with the start of puberty. So before puberty start, these Spermatogonia A, Pale and Dark, they keep on sitting in these recesses of Sertoli cells. They are not doing anything, they are resting there, right, as such. Now, how do they start spermatogenesis? What happens is that as the brain matures, before puberty start, as the brain matures, through hypothalamus releases a special substance that is called GnRH, gonadotropin releasing hormone. This gonadotropin releasing hormone, it will go into anterior pituitary, and here it will cause synthesis of two substances, synthesis and release of two substances. What are those? FSH and LH, follicle stimulating hormone and luteinizing hormone, right? So luteinizing hormone, it goes to Leydig cells. What are Leydig cells? Outside these Sertoli cells and outside the seminiferous tubules, in the connective tissue, in the interstitium, this Leydig cells are present and blood vessels are there, right? And loose connective tissue is there, right? So luteinizing hormone will go to Leydig cell and it will stimulate the Leydig cell to synthesize testosterone. What it will cause? It will cause synthesis of testosterone hormone. Most of you must be aware of role of this luteinizing hormone in female reproductive system. But you also must know that this luteinizing hormone has a very important role in males as well. Its Leydig cells, luteinizing hormone stimulates Leydig cells, right? Alpha Leydig, alpha luteinizing hormone. I like to call it Leydig hormone as well. Well, it is not its official name, but it has a more descriptive name in males. We also call it interstitial cell stimulating hormone, because Leydig cell, it is present in interstitium, right? So it is also interstitial cell. Leydig cell is interstitial cell, and this luteinizing hormone, because it stimulates this interstitial cell, we also like to call it interstitial cell stimulating hormone in males, right? So this is Leydig hormone. So testosterone will be released, synthesized and released, and most of this testosterone will go into blood vessel and will go into peripheral blood, and it will cause what it will cause? It will cause development of secondary sexual characteristics in male, development of beard, muscles, all that things, pubic hair, right? All that thing. And some of the testosterone, it will also go into, what is this cell? This Sertoli cell, right? And I will tell you what it will do. And then there is FSH as well, follicle stimulating hormone. It will also go towards Sertoli cells. Here it has extracellular receptors. And now testosterone as well as follicle stimulating hormone, they will stimulate, what is this, Sertoli cell, and they will tell the Sertoli cell that it is time, puberty has started. Now it is the time to start synthesis of sperms, right? So Sertoli cell will release special growth factors that will start the process of spermatogenesis. And that's how spermatogenesis starts. I hope it is not too complicated for you. Clear? So under the influence of growth factors, spermatogenesis will start. Now let's remove the Sertoli cell and all these things and just look at what happens with these cells. So here we have, what are these? Spermatogonia A, Pale and Dark. Forget about Dark, you know its function. Let's talk about Spermatogonia A Pale. So under the influence of growth factors, after multiple cell division, it will form this cell, and it has also the ability of self renewal. It will also maintain its own population. And what is this cell? It is Spermatogonia B. So Spermatogonia A will ultimately form a more specialized cell, that is Spermatogonia B. Spermatogonia B is not stem cell. It cannot undergo, it has not the ability of self-renewal, and it is more specialized form, and it is called Spermatogonia B. Now what will happen? Spermatogonia B is still not ready for meiosis. It needs to be further differentiated. It will form a more differentiated cell. And what is that more differentiated cell? Through mitosis, it will form primary spermatocyte. And this primary spermatocyte, it is now ready for meiosis. It is not this cartoon, it is actually cell. So let's make it like cell. I hope you do not tell your teacher in Viva that it is that cartoon that undergo meiosis, right? So primary spermatocyte will be formed. And now this primary spermatocyte will undergo meiosis to form mature sperm. But now there is a problem here. What is the problem? The problem is that when this primary spermatocyte will undergo meiosis, right, and it will undergo crossing over, and number of chromosomes will be halved, what will happen is that the DNA of that subsequent cells, it will be different from all those cells of the body. So genetic makeup will be quite different from all other cells of the body. So if immune system of our body, when it came across such cell that has different genetic makeup than other cells of the body, it may consider it as a harmful substance. So immune system may try to destroy those cells that had underwent meiosis. So Sertoli cells, these are also called nerve cells or mother cells or sporting cells or sustentacular cells, right? They will come here to rescue. So here Sertoli cell, they have these cytoplasmic projections or cytoplasmic processes, and these processes, they are closely linked with each other with the help of special proteins or tight junctions. Now primary spermatocyte will have to cross that junction and they will come into here, this area. So it will provide this special microenvironment in which it can undergo meiosis, and it will be hidden from immune system. You see this diagram that here they will come here at this place, and here these cells, they are prevented from here, these cells, they are hidden from the immune system, right? This is blood testis barrier. So how this primary spermatocyte will come into this hidden area to hide themselves from the immune system? Will this blood testis barrier open up for it? No, it will not. That is a dangerous thing. If it opens up even for a microsecond, some immune cells may go into here. Some cytotoxic substance may go into this microenvironment. So nature has provided a foolproof system here. What is that foolproof system here? Let's look at the animation. What will happen that as it moves forward, a bulge is formed and another cytoplasmic projection, another cytoplasmic process is formed here, and another blood testis barrier is formed here, right? So now we have two barriers, one that was previously present and a new process is formed here, a new barrier is formed here, right? And then the old barrier will be removed, right? And now it is in this hidden area, right? So look at this beautiful foolproof method that nature has provided. So now primary spermatocyte, they are hidden from immune system, and now they can happily undergo meiosis 1 to form secondary spermatocyte, and then secondary spermatocyte, this is meiosis 1, and then secondary spermatocyte will undergo meiosis 2 to form spermatid. Remember, the secondary spermatocyte, they are half the size of primary spermatocyte, and spermatid is half the size of secondary spermatocyte, right? And this then spermatid, they will undergo cytodifferentiation to form mature sperm. We will discuss in next lecture, and that process is called spermiogenesis. So next we will discuss what is spermiogenesis. Here I would like to highlight few important points. Number one, you must have noticed that these cells, they are not fully separated from each other, right? You can see there are these bridges that are present here. So why bridges are there? Why these gaps are there in between these cells? Why they are still connected to each other? So actually what happens that when they undergo cell division, cytokinesis, you know what is cytokinesis? Cytokinesis is that when cytoplasm separates and it form two different cells, right? They are separated from each other. So cytokinesis is incomplete here. So not what happens that cytokinesis is not complete, so they remain attached at this point, right? Why these bridges are present? You know why they are present? Well, the reason is simple. These Sertoli cells, they have to provide growth factors and nourishment to millions, rather billions of these developing sperms, right? So it is not efficient method. Economically, it is not efficient method to provide growth factor to each and every developing spermatocyte, each and every developing sperm, right? So because gaps are there, if Sertoli cell provide growth factor only to this cell, right, it will automatically, these growth factors, they will automatically diffuse their effects to neighboring cells as well, because they are connected to each other. So look at this economicity here, right? How beautiful nature has provided this mechanism that it can provide nourishment and growth factors. They can diffuse from one cell to neighboring cell, right? So that is the reason. When I was kid, when I was a medical student, I had a very seemingly dumb question that I like, that I hid from my class fellows that they should not mock me. Well, I think there is no dumb question. So the question is that why these primordial germ cells are there? Why not this mature sperm, they copy itself? Why not mature sperm, they undergo mitosis? One mature sperm undergo mitosis to form two, two into four, and so on. Why they start from the bottom, from primordial germ cell and from rather spermatogonia? The reason is simple. Number one, each and every sperm is different, right? Each and every sperm is different from its brother, right? The other sperm. Why it is different? Due to meiosis, due to crossing over. So if we copy the sperm, then all of your siblings, they will look quite similar to each other, right? The other reason is that as a cell matures, as a cell becomes more specialized, it is difficult to copy that cell. It is difficult for that cell to divide itself. Why? The reason is that as a cell matures, as a cell becomes more specialized, most of its genes are turned off. Most of its DNA, most of its genetic material is more coiled. It becomes tightly coiled. It becomes heterochromatin, and only those genes are loosely arranged, or euchromatin we call them. Only those genes are loosely arranged that has to perform essential functions for that cell, right? So it is very difficult for a mature sperm to copy itself. And that is not true for the just sperm. It is true for skin cells. It is true for any other cells, for the GIT epithelial cells. That is why nature has provided us with adult stem cells. For example, basal cells in the skin, right? They will form ultimately a mature epithelial cell, or whatever cell that is required, right? So this was about spermatogenesis. Next we will discuss spermiogenesis. If you like this video, please press the thumbs up button. Please subscribe to my channel if you haven't already, and please support me on Patreon. Also, please give your precious comments about this lecture. Thank you so much.